Hello and welcome to pH Biology. Today's explainer is the eukaryotic cell. If a cell has a nucleus enclosing its DNA, it's eukaryotic. That puts plants, animals, fungi and protists in one big family. By the end of this video, you'll know exactly what defines a eukaryote, the organelles to expect inside one, and the single key difference that separates eukaryotes from bacteria. A eukaryotic cell has a nucleus, a membrane-bound compartment that contains the DNA. It also has other membrane-bound organelles, mitochondria, often chloroplasts in plants, an endoplasmic reticulum, Golgi apparatus, and so on. Eukaryotic cells are typically 10 to 100 times larger than prokaryotic cells. Animals, plants, fungi, and protists are all built from eukaryotic cells. 3 things to lock in. 1. DNA is sealed in a nucleus, not floating in the cytoplasm. 2. There are membrane-bound organelles like mitochondria for respiration and chloroplasts for photosynthesis. And 3. Size, typically 10 to 100 micrometers, much bigger than bacteria. If a cell ticks all 3 boxes, it's eukaryotic. The classic slip is confusing eukaryotic and prokaryotic. Eukaryotic has a nucleus, prokaryotic doesn't, and bacteria are not eukaryotic, they're prokaryotic. The presence or absence of a nucleus is the single most important distinguishing feature when you're asked to identify a cell type in an exam. Quick example. A typical animal cell, nucleus, mitochondria, ribosome, cell membrane. A typical plant cell, all of the above, plus chloroplasts, a cell wall, and a permanent vacuole. Both are eukaryotic. So is a yeast cell or a paramecium. Anything with a nucleus, you're in eukaryotes territory. Right, if any of this... Hello and welcome to pH Biology. Today's explainer is the prokaryotic cell. No nucleus. DNA floating loose in the cytoplasm. Tiny. By the end of this video, you'll know what makes a cell prokaryotic, why plasmids matter for biotechnology, and exactly how prokaryotes differ from eukaryotes on the GCSE paper. A prokaryotic cell has no nucleus. Its DNA sits free in the cytoplasm, often joined by small extra circles called plasmids. There are no membrane-bound organelles like mitochondria or chloroplasts. Prokaryotic cells are tiny, typically 1 to 10 micrometers, 10 to 100 times smaller than eukaryotes. Bacteria are the textbook example of a prokaryotic cell. Three things to remember. 1. There's no nuclear membrane, so the DNA is naked in the cytoplasm. 2. Generally no membrane-bound organelles. And 3. They're tiny. They do have cell walls, ribosomes, sometimes flagella for movement, and plasmids that can be transferred between bacteria, which is exactly why genetic engineering uses them. Common mistake. Prokaryotes definitely have DNA. They just don't enclose it in a nucleus. And don't confuse prokaryotic with simple. These cells have complex chemistry, including the ability to mass produce human insulin when we engineer their plasmids in the lab. Quick example, E. coli is a typical prokaryote. A single cell, no nucleus, plasmid DNA, surrounded by a cell wall. We use E. coli extensively in biotechnology. Insert a human gene into a plasmid, put it back into the bacterium, and the bacterium becomes a tiny factory producing human protein. Hello and welcome to pH Biology. Today's explainer is magnification. How many times bigger the image is than the real thing. By the end of this video, you'll have the formula locked in, you'll know how to convert units correctly, and you'll calculate magnification problems in the GCSE paper without losing a mark. Magnification equals image size divided by actual size. The same units on the top and bottom always. That's the rule. If you've got the image in millimeters and the actual size in micrometers, you have to convert one before dividing. Magnification has no units. It's just a number, written as times something. Three things to lock in. 1. Convert all measurements to one common unit before dividing. 2. The conversion. 1 millimeter is 1,000 micrometers, which is 1 million nanometers. 3. When you use both an eyepiece and an objective lens, the total magnification is the eyepiece magnification times the objective magnification. The classic slip is forgetting to convert. If the image is 5 centimeters and the actual size is 50 micrometers, you cannot just divide 5 by 50. You have to convert 5 centimeters into 50,000 micrometers and then divide. Always check that both numbers are in the same unit before you press the calculator button. Quick example. Image size 5 centimeters, which is 50,000 micrometers. Actual size 50 micrometers. Magnification equals 50,000 divided by 50, which is times 1,000. Two-line method, full marks, convert, divide, done. Hello and welcome to pH Biology. Today's explainer is resolution, the smallest detail a microscope can show you. By the end of this video, you'll know exactly what resolution means, why it's different from magnification, and why electron microscopes can show structures that light microscopes simply cannot. Resolution is the minimum distance between 2 points that can still be distinguished as separate. If two structures are closer than the resolution, they blur into one. Resolution is limited by the wavelength of the radiation used to illuminate the specimen. Shorter wavelength means higher resolution and finer detail. 3 things. 1. Light microscopes have a resolution of around 200 nanometers. 2. Electron microscopes go as low as 0.2 nanometers, 1,000 times better. And 3. The electron microscope wins because electrons have much shorter wavelengths than visible light. Higher resolution lets you see smaller things in genuine detail. Common slip. Don't confuse resolution with magnification. Magnification makes things appear bigger. Resolution decides what you see as sharp or just a magnified blur. A useful microscope needs both. High magnification with low resolution just gives you a giant smudge. Quick example. 2 viruses sitting 100 nanometers apart. Under an electron microscope with 0.2 nanometers resolution, they appear as 2 clearly separate viruses. Under a light microscope with 200 nanometers resolution, they merge into a single blur. Same viruses, different stories. Hello and welcome to pH Biology. Today's explainer is diffusion, how particles spread out from where there are lots to where there are fewer. By the end of this video, you'll know the precise definition, the 3 factors that change the rate, and exactly where diffusion matters in your body right now. Diffusion is the net movement of particles from a region of higher concentration to a region of lower concentration down the concentration gradient. No energy is required. It's driven by random thermal motion of particles. The word net is important. Particles move both ways, but more flow from where there's more to where there's less. 3 things to lock in. 1. Steeper concentration gradient gives a faster rate of diffusion. 2. Higher temperature means particles move faster and the rate goes up. 3. Larger surface area gives more places for particles to cross and increase the rate. Adapt 1, 2, or all 3 to speed it up. Common slip. Diffusion is the net movement. Particles move both ways across a membrane. We describe the overall direction. And don't confuse diffusion with osmosis. Diffusion is for any small particle. Osmosis is specifically the movement of water across a partially permeable membrane. Quick example, in the alveoli of your lungs, oxygen is at a high concentration in the air and low concentration in the blood. Oxygen diffuses from alveoli into blood, down the gradient. Carbon dioxide does the opposite, diffusing from blood into the alveoli, ready to be exhaled. Hello and welcome to pH Biology. Today's explainer is osmosis, water moving through a membrane to balance the solute. By the end of this video, you'll know the proper definition, what happens to plant and animal cells in different solutions, and why osmosis runs every required practical with potato cylinders. Osmosis is the net movement of water from a region of higher water potential, a more dilute solution, to a region of lower water potential, a more concentrated solution, across a partially permeable membrane. Only water moves. Solute particles cannot pass through, and no energy is required. Three things. 1. The membrane must be partially permeable. 2. Water moves down its water potential gradient, which means into the more concentrated solution. 3. A plant cell in pure water becomes turgid. An animal cell in pure water bursts because it has no cell wall to limit expansion. Common slip. Osmosis is about water specifically, not any liquid, and the direction can confuse. Water moves to where there's more solute. A potato cylinder placed in concentrated sugar solution loses mass. Water is leaving the cells through osmosis into the surrounding solution. Quick example. A potato cylinder in distilled water gains mass. Water enters cells by osmosis. Potato cylinder in concentrated sugar solution loses mass. Water leaves the cells by osmosis. Plot percentage change in mass against sugar concentration, and where the line crosses 0 is the water potential of the potato cells. Hello and welcome to pH Biology. Today's explainer is active transport, pumping particles uphill against the concentration gradient using energy from respiration. By the end of this video, you'll know what it makes it different from diffusion, why it needs mitochondria, and exactly where it happens in your body. Active transport is the net movement of particles from a region of lower concentration to a region of higher concentration against the concentration gradient. This requires energy in the form of ATP from respiration. Carrier proteins in the membrane change shape, pumping the particles through. It's the opposite direction to diffusion. Three things. 1. It always works against the gradient. 2. It needs energy from respiration, supplied as ATP from mitochondria. 3. Cells that do lots of active transport have many mitochondria. Root hair cells, intestinal lining cells, kidney cells. Spot lots of mitochondria, expect active transport. Common slip. Don't say down the gradient, because that's diffusion. Active transport is the opposite. And don't forget the role of mitochondria. They supply the ATP that powers the carrier proteins. No mitochondria, no ATP, no active transport. Quick example, root hair cells absorb mineral ions from soil even though the ions are at low concentration in soil and high concentration in the root. Active transport pumps them in against the gradient, using ATP from the root hair cells' many mitochondria. Same trick in the small intestine. Glucose absorbed against gradient. Hello and welcome to pH Biology. Today's explainer is mitosis, one cell dividing into two genetically identical daughter cells. By the end of this video, you'll know the 4 stages, why the daughter cells are identical, and exactly when mitosis happens in your body. Mitosis is nuclear division producing 2 genetically identical diploid daughter cells from one parent cell. It happens after DNA has replicated, ensuring each new cell gets a full copy. Mitosis is used for growth, repair, and asexual reproduction. Gametes, the sperm and eggs, are made by a different process called meiosis. Memorize PMAT: prophase, metaphase, anaphase, telophase. Prophase, the chromosomes condense, nuclear membrane breaks down, a spindle forms. Metaphase, chromosomes line up at the equator. Anaphase, sister chromatids are pulled to opposite poles. And the telophase, chromosomes decondense, new nuclear membranes form. Then cytokinesis splits the cytoplasm into 2 daughter cells. Common slip. Mitosis is for body cells. Gametes are made by meiosis, which halves the chromosome number. Mitotic daughter cells are diploid, full chromosome count, and identical to the parent. Mitosis happens constantly in your body. Skin replacing itself, bone healing a fracture, embryo developing from a fertilized egg. Quick example, a fertilized egg divides by mitosis to produce every cell in your body. After just 5 mitotic divisions, you have 2 to the 5, which is 32 cells. By the time you're born, you're made of trillions, every one a descendant of mitotic division from that original zygote. Hello and welcome to pH Biology. Today's explainer is the enzyme, a biological catalyst that speeds up the chemical reactions of life. By the end of this video, you'll know what enzymes are made of, why they're specific, and the 3 digestive enzymes you must know cold. An enzyme is a biological catalyst. It speeds up a chemical reaction without being used up. Enzymes are proteins with a specific 3-dimensional shape that includes an active site where the substrate binds. Each enzyme catalyzes one specific reaction. Enzymes are reusable. The same molecule catalyzes thousands of reactions in its lifetime. Three things. 1. Enzymes are proteins, so their shape is everything. 2. Each enzyme is specific to one substrate, like a lock and single key. 3. Enzymes work at body temperature without high heat because they lower the activation energy of the reaction they catalyze. Common slip. Enzymes are not alive. They don't die. The proper word is denature, when the shape of the active site is destroyed by heat or extreme pH. And don't say enzymes work on all foods. Each enzyme catalyzes one specific reaction. Choose the wrong one and nothing happens. Quick example. Amylase breaks down starch into the sugar maltose. Protease breaks down proteins into amino acids. Lipase breaks down lipids into fatty acids and glycerol. 3 enzymes, 3 substrates, 3 sets of products. Same general idea. Bring the substrate to the active site, break the bonds. Hello and welcome to pH Biology. Today's explainer is the active site, the small pocket on an enzyme where the substrate binds. By the end of this video, you'll know what shapes the active site, why specificity matters, and exactly what heat does to it. The active site is a specific region on an enzyme where the substrate binds. It's shaped to fit one particular substrate, like a lock for a key. The active site shape is held in place by hydrogen bonds and ionic interactions in the protein's tertiary structure. Disrupt those bonds and the active site loses its shape. Three things. 1. The active site is shaped by the protein's tertiary structure. 2. Heat or extreme pH disrupts the bonds and the active site changes shape. 3. Once denatured, the substrate no longer fits. The reaction stops, often permanently. This is the heart of why enzymes have an optimum temperature. Common slip. Don't think of the active site as eating the substrate. The substrate binds in the active site. The reaction happens and the products are released. The enzyme is unchanged and ready to bind another substrate molecule. Once a million reactions per second in some cases. Quick example, amylase has an active site shaped to fit starch. Heat amylase above 60 Celsius and the bonds holding the active site break. The pocket changes shape, starch no longer fits, and the amylase stops working. That's why we keep the amylase pH practical at 35, to keep the active site intact. Hello, welcome to pH Biology. Today's explainer is denaturation, the change in shape of an enzyme caused by heat or extreme pH. By the end of this video, you'll know what causes it, why it's usually permanent, and how it shows up on every enzyme rate graph. Denaturation is a change in the shape of an enzyme, particularly its active site, caused by heat or extreme pH. The bonds that hold the protein in its three-dimensional shape, especially hydrogen bonds and ionic interactions, break. The active site no longer fits the substrate and the enzyme stops catalyzing the reaction. Three things. 1. Denaturation is usually permanent. Cooling a denatured enzyme back to body temperature doesn't restore the active site shape. 2. Different enzymes denature at different temperatures and pH values. 3. On a rate temperature graph, denaturation appears as the sharp drop in rate above the optimum. Common slip. Say denatured, not died or killed. Enzymes aren't alive, and don't say the enzyme is broken. The amino acids are still in the right sequence, but the three-dimensional shape has changed. Without that shape, the enzyme is useless. Quick example, frying an egg. The clear gel of egg white is mostly the protein albu, albumin in solution. Heat the egg and the albumin denatures. The protein unfolds, tangles with neighbors, and the clear liquid becomes opaque and solid. Same molecular process as denaturing a digestive enzyme. Hello and welcome to pH Biology. Today's explainer is the lock and key model of enzyme action. One key per lock, one substrate per enzyme. By the end of this video, you'll know why this model explains enzyme specificity, when to use it in an exam, and where it falls short for higher study. The lock and key model says the substrate fits the active site of the enzyme like a key fits a specific lock. The substrate must be the right shape to bind. Once bound, the reaction happens and the products are released. The enzyme is unchanged. Only the correct substrate shape can fit each enzyme, which is why enzymes are specific. 3 things. 1. The substrate shape must match the active site shape. 2. Only one type of substrate can bind to any one enzyme. This is enzyme specificity. 3. After the reaction, the products are released and the enzyme is free to bind another substrate molecule. It's a simple model, but very useful. Common slip. At GCSE, stick with lock and key unless you're asked otherwise. At A level, you'll meet the more flexible induced fit model, where the active site adjusts slightly as the substrate binds. But for GCSE, the rigid lock and key is the expected answer. Quick example, glucose fits the active site of hexokinase, an enzyme that adds a phosphate to it during respiration. Fructose, which is almost the same shape, does not fit hexokinase. That tiny shape difference is enough to make the enzyme reject it. That's what enzyme specificity really means. Hello and welcome to pH Biology. Today's explainer is bile, a green liquid made in the liver that does 2 crucial jobs in digestion. By the end of this video, you'll know exactly what bile does, where it's made and stored, and why it isn't an enzyme at all. Bile is a liquid made in the liver and stored in the gallbladder. It's released into the small intestine when fatty food arrives. Bile has 2 functions. It emulsifies lipids, breaking big fat droplets into many small ones, and it's alkaline, neutralizing the acid chyme coming from the stomach. Three things. 1. Bile emulsifies fats by breaking droplets up, vastly increasing surface area for lipase to attack. This is a physical process. And 2. Bile is alkaline, neutralizing stomach acid so the small intestine can operate at its optimum pH of around 8. 3. Both effects help digestion proceed efficiently. Common slip. Bile is not an enzyme. It doesn't chemically digest anything. Lipase digests fat. Bile just makes lipase's job easier by increasing the surface area available. And remember, liver makes bile. Gallbladder stores it. 2 different organs with 2 different roles. Quick example. After a fatty meal, bile is released into the small intestine. The fat droplets are broken into thousands of small droplets. Picture a salad dressing being shaken. The total surface area goes up dramatically, and lipase can then digest the fats much faster than before. Hello, and welcome to pH Biology. Today's explainer is villi, tiny finger-like projections lining the small intestine. By the end of this video, you'll know their structure, their function, and why the small intestine is one of the best adapted surfaces in the human body. Villi are finger-like projections lining the small intestine that vastly increase the surface area for absorption of digested food. Each villus is one cell thick on the outside, providing a short diffusion path. It contains a network of capillaries and a lacteal, a lymphatic vessel, to carry the absorbed nutrients away. Three things. 1. Villi increase surface area. Microvilli on each villus cell increases it even further. 2. The one cell thick wall gives a short diffusion path. 3. The rich capillary network removes absorbed nutrients quickly, maintaining a steep concentration gradient between gut and blood, which keeps absorption fast. Common slip. Villi don't digest food. Digestion happens before the food reaches them. Villi absorb the products of digestion, glucose, amino acids, fatty acids. Many of those absorptions use active transport, not just diffusion, needing ATP from the mitochondria packed into the villus cells. Quick example, without villi, the small intestine wall would have a surface area of around half a square meter. With villi and microvilli, the same length of gut provides around 250 square meters, the size of a doubles tennis court. That's how a tube 1 centimeter wide absorbs your entire breakfast. Hello and welcome to pH Biology. Today's explainer is amylase, the enzyme that breaks starch into the sugar maltose. By the end of this video, you'll know exactly what amylase does, where it's produced, and the trick that makes the required practical work. Amylase is a digestive enzyme, a carbohydrase, that breaks starch into the sugar maltose. It's produced in the salivary glands of the mouth, in the pancreas, and in the lining of the small intestine. Optimum pH is around 7, so amylase works in the mouth and the small intestine, but not in the stomach where the pH is 2. Three things. 1. Substrate is starch, product is maltose. 2. Optimum pH around 7, denatures in the very acidic stomach. 3. Used in the famous required practical to measure rate versus pH, using iodine to detect when all the starch has been digested. Iodine plus starch turns blue-black. Common slip. Amylase only works on starch. Don't confuse it with maltase, which breaks maltose into glucose. And note the pH. In the stomach, amylase is denatured by the acid, so digestion of starch pauses there and resumes in the small intestine. Quick example. Chew a cracker slowly for 30 seconds. After a while, you'll notice a sweet taste. That's amylase from your saliva breaking the cracker's starch into maltose. Without chewing thoroughly, you get less amylase exposure, and digestion of the starch begins later in the gut. Hello and welcome to pH Biology. Today's explainer is photosynthesis, the process that builds every bit of plant matter on Earth from carbon dioxide, water, and sunlight. By the end of this video, you'll know both equations, the site where it happens, and why it's endothermic. Photosynthesis is the process by which plants make glucose from carbon dioxide and water using light energy absorbed by chlorophyll. Word equation: carbon dioxide plus water gives glucose plus oxygen in the presence of light. Symbol equation: 6CO2 plus 6H2O gives C6H12O6 plus 6O2. It's endothermic because it needs energy in. Three things. 1. Light energy is absorbed by chlorophyll, the green pigment in chloroplasts. 2. Photosynthesis happens in chloroplasts, mostly in the palisade mesophyll cells of leaves. 3. The glucose produced is used immediately for respiration, converted to starch for storage, or used to build cellulose and other plant molecules. Common slip. Say glucose, not just food. Photosynthesis specifically produces glucose, the simple sugar that everything else is built from. And remember, oxygen is a byproduct. It's released through stomata into the atmosphere, but the plant didn't set out to make it. Quick example. 6 CO2 plus 6 H2O gives C6H12 C12O6 plus 6 O2. Balance check. 6 carbons in, 6 in glucose, 12 hydrogens in, 12 in glucose, 18 oxygens in, 6 in glucose, and 12 in oxygen molecules. Total 18 out. Balance perfectly. Every single oxygen we breathe ultimately came from photosynthesis. Hello and welcome to pH Biology. Today's explainer is chlorophyll, the green pigment that captures light energy for photosynthesis. By the end of this video, you'll know what color it absorbs, why leaves look green, and what happens when chlorophyll is missing. Chlorophyll is the green pigment in chloroplasts that absorbs light energy for photosynthesis. It absorbs mostly red and blue wavelengths of visible light while reflecting green, which is why leaves appear green to our eyes. Chlorophyll is essential. Without it, no photosynthesis and no plant could survive. Three things. 1. Chlorophyll is a pigment, not an enzyme. 2. It lives inside chloroplasts in plant cells, mostly in the palisade mesophyll cells. And 3. Absorbing red and blue, reflecting green, is why plants look green. In autumn, chlorophyll breaks down and the other pigments, yellow, orange, and red, become visible. Common slip. Chlorophyll isn't the chloroplast. Chlorophyll is the pigment. The chloroplast is the organelle housing it. And chlorophyll isn't the only photosynthetic pigment. There are carotenoids too, but at GCSE you focus on chlorophyll. Quick example. Grow 2 seedlings, one in red light, one in green light. The red light seedling thrives. Chlorophyll absorbs red wavelengths and photosynthesis runs fast. The green light seedling struggles. Chlorophyll reflects green, so very little light is absorbed and photosynthesis is slow. Hello and welcome to pH Biology. Today's explainer is limiting factors. The slowest supplying factor sets the rate of photosynthesis. By the end of this video, you'll know what counts as a limiting factor, how to spot one on a graph, and why greenhouse growers care all about this. A limiting factor is the factor that's currently restricting the rate of photosynthesis. Increasing it would speed up photosynthesis, while increasing other factors would have no effect. The most common limiting factors are light intensity, carbon dioxide concentration, temperature, and the amount of chlorophyll. Three things. 1. Only one factor is limiting at any moment. 2. When a graph plateaus, the limiting factor has changed to something else. 3. At low carbon dioxide, increasing carbon dioxide raises the rate proportionally. At high carbon dioxide, the rate plateaus because something else, like light, temperature, or chlorophyll, is now limiting. Common slip. When a rate graph plateaus, photosynthesis hasn't stopped. It's still happening, just at a steady rate limited by some other factor. Greenhouse growers exploit this by controlling all three potentially limiting factors at once: artificial light, extra carbon dioxide, and heating. Quick example, a graph of photosynthesis rate against carbon dioxide concentration at low light shows a quick plateau. Light becomes limiting. The same graph at high light shows a higher plateau. Carbon dioxide becomes limiting at a higher level. Match the rate curve to which factor is limiting. Hello and welcome to pH Biology. Today's explainer is aerobic respiration. Glucose plus oxygen gives carbon dioxide plus water, releasing the most energy possible from each glucose molecule. By the end of this video, you'll know both equations, the site and what aerobic actually means. Aerobic respiration is the process by which cells release energy from glucose using oxygen. Word equation: glucose plus oxygen gives carbon dioxide plus water with energy released. Symbol equation: C6H12O6 plus 6O2 gives 6CO2 plus 6H2O. The reaction is exothermic. It happens in mitochondria. Three things. 1. Aerobic respiration uses oxygen. 2. It releases lots of energy per glucose as ATP. 3. Every living cell respires. Cells with high energy demands, muscle cells, intestinal cells, root hair cells, contain many mitochondria. Spot lots of mitochondria, expect lots of aerobic respiration. Common slip. Respiration is not breathing. Breathing is ventilation, moving air in and out of the lungs. Respiration is the chemistry happening inside every living cell, releasing energy from glucose. Plants respire too, day and night, even when not photosynthesizing. Quick example, you inhale oxygen, exhale carbon dioxide and water vapor. The carbon and hydrogen in that carbon dioxide and the water vapor came from the glucose you ate. The oxygen you inhaled combined with that glucose, releasing the energy your cells need to keep working. Hello and welcome to pH Biology. Today's explainer is anaerobic respiration, respiration without oxygen, releasing less energy than aerobic, with different products in animals and yeast. By the end of this video, you'll know both forms, when each happens, and what causes that burning feeling in your legs at the end of a sprint. Anaerobic respiration releases energy from glucose without oxygen, and there's two forms. In animal muscle cells, glucose gives lactic acid. In yeast cells, glucose gives ethanol and carbon dioxide, which is called fermentation. Both produce less energy per glucose than aerobic respiration because the glucose isn't fully broken down. Three things. 1. Animals make lactic acid. Yeast makes ethanol plus carbon dioxide. 2. Far less energy released because glucose isn't fully oxidized. Most energy stays in the lactic acid or ethanol. And 3. Lactic acid is acidic and lowers the pH of muscle tissue, causing cramp and fatigue during heavy exercise. Common slip. Anaerobic in plants and yeast is fermentation with ethanol and carbon dioxide. Anaerobic in animal muscle is lactic acid only. Don't mix them up. And remember, anaerobic respiration is not better than aerobic. It's just a fallback when oxygen can't be supplied fast enough. Quick example. Sprint 100 meters flat out. Your legs can't take in oxygen fast enough. Muscle cells switch to anaerobic respiration. Lactic acid builds up, your muscles ache, and at the end you keep panting to repay the oxygen debt, getting rid of the lactic acid. Hello and welcome to pH Biology. Today's explainer is oxygen debt, the extra oxygen the body needs after exercise to break down the lactic acid that built up during anaerobic respiration. By the end of this video, you'll know exactly what it is, how the body repays it, and why you pant after a sprint. Oxygen debt is the extra oxygen needed after exercise to break down the lactic acid that built up during anaerobic respiration. While you're sprinting, your muscles outpace oxygen supply and switch to anaerobic respiration, producing lactic acid. After you stop, your body still needs oxygen to clear the lactic acid. 3 things. 1. Heart rate and breathing rate stay high after exercise to deliver the extra oxygen. 2. Lactic acid is oxidized to carbon dioxide and water, or transported to the liver and converted back to glucose. And 3. The bigger the anaerobic effort, the bigger the debt to repay. Common slip. Oxygen debt isn't lack of oxygen during exercise. That's just anaerobic respiration. Oxygen debt specifically refers to the extra oxygen needed after exercise has stopped. Even after you've stopped running, the debt is still there until the lactic acid is fully broken down. Quick example. Run a 100-meter sprint. Let's say total race time 12 seconds, but you keep panting for another 2 minutes after the finish line. That extra panting is repaying the oxygen debt, supplying the oxygen needed to clear the lactic acid your muscles produce during the sprint. Hello and welcome to pH Biology. Today's explainer is fermentation. Anaerobic respiration in yeast producing ethanol and carbon dioxide. By the end of this video, you'll know why we use it in baking and brewing and what's actually happening when the bread dough rises. Fermentation is anaerobic respiration in yeast. Glucose gives ethanol plus carbon dioxide. No oxygen needed. Yeast is a single-celled fungus that ferments sugars naturally when oxygen is scarce. Less energy released per glucose than aerobic respiration, but the byproducts are exactly what we want for baking and brewing. Three things. 1. Used in baking. Carbon dioxide makes dough rise. Ethanol evaporates during baking. 2. Used in brewing and winemaking. Ethanol stays and gives the alcoholic content. 3. Fermentation is anaerobic, happens when oxygen is unavailable. Add lots of oxygen and yeast switches to aerobic respiration instead. Common slip. Fermentation in yeast gives ethanol and carbon dioxide. In animal muscle, anaerobic gives lactic acid. Different process, different products. And remember, yeast is alive. It's a fungus, not just a chemical reagent. That's why it dies if conditions are wrong. Quick example, mix yeast with flour and warm water plus a little sugar. Within minutes, bubbles of carbon dioxide form, dough rises, and you can faintly smell alcohol. Bake the dough. Carbon dioxide bubbles fix in place, ethanol evaporates, yeast dies. You're left with risen bread. Hello and welcome to pH Biology. Today's explainer is the compensation point, the moment when a plant's photosynthesis exactly matches its respiration and there's no net gas exchange with the air. By the end of this video, you'll know what's happening and when it occurs. The compensation point is the light intensity at which a plant's rate of photosynthesis exactly equals its rate of respiration. The plant produces oxygen at the same rate it uses it. It consumes carbon dioxide at the same rate it releases it. There is no net exchange of gases with the environment. 3 things. 1, both processes are happening simultaneously. They don't stop, they just cancel out. 2, below the compensation point, respiration outpaces photosynthesis, so the plant is a net consumer of oxygen. 3, above the compensation point, the plant is a net producer of oxygen. Common slip. At the compensation point, photosynthesis hasn't stopped. It's just equal to respiration. And remember, plants respire day and night. They only photosynthesize in the light. The compensation point is where the two processes are exactly equal in rate. Quick example, at dawn or dusk, light intensity is low. If light is just enough for photosynthesis to match the plant's constant rate of respiration, you've reached the compensation point. Lower light, plant uses more oxygen than it makes. Higher light, plant becomes a net producer of oxygen. Hello and welcome to pH Biology. Today's explainer is DNA, the double helix molecule that stores the genetic instructions for every living organism. By the end of this video, you'll know the bases, the pairing rules, and what makes a gene a gene. DNA stands for deoxyribonucleic acid. It's a long molecule arranged as a double helix, 2 strands wound around each other like a twisted ladder. The rungs of the ladder are made of 4 bases, A, T, G, and C. A pairs with T, G pairs with C. The base pairing is the basis of inheritance. 3 things. 1, bases pair specifically, A with T, G with C. 2, a gene is a section of DNA that codes for a particular protein. 3 bases code for 1 amino acid. So a gene with 360 bases codes for a protein with 120 amino acids. Common slip. DNA isn't a chromosome. A chromosome is condensed DNA wrapped around proteins, visible during cell division. And the 2 strands of the double helix run in opposite directions, anti-parallel, though this detail is more A-level than GCSE. Quick example, a 360 base gene divides into 120 triplets of bases. Each triplet codes for 1 amino acid, so the gene produces a protein made of 120 amino acids. Change 1 base, a mutation, and you might change 1 amino acid in the protein. Hello and welcome to pH Biology. Today's explainer is the gene, a section of DNA that codes for a particular protein. By the end of this video, you'll know what genes are, where they sit, and how they determine your characteristics. A gene is a length of DNA on a chromosome that codes for a particular protein, or more specifically, a particular sequence of amino acids that fold to make a protein. The protein then determines a characteristic. Different versions of the same gene are called alleles. You inherit 1 copy from each parent. 3 things. 1, the order of bases in a gene determines the order of amino acids in the protein. 2, the protein determines the characteristic. 3, each gene is at the same position on the chromosome in everyone, but different people may have different alleles of that gene. Common slip. A gene is a section of DNA, not a whole chromosome. And don't say a gene is a protein. A gene codes for one. The gene is the recipe. The protein is the dish. The recipe doesn't taste of anything, only the dish does. Quick example, the cystic fibrosis gene codes for a protein that transports chloride ions across cell membranes. A mutation in this gene changes the protein's amino acid sequence. The protein folds wrong, and chloride transport fails. Sticky mucus builds up. That single gene determines whether someone has cystic fibrosis. Hello and welcome to pH Biology. Today's explainer is the allele, different versions of the same gene. By the end of this video, you'll know what makes an allele different from a gene, how many alleles you have for each gene, and why alleles matter for inheritance. An allele is a different form of the same gene. The gene is at a fixed position on the chromosome, the same position in everyone, but the gene can come in different versions. Each version is an allele. You inherit 2 alleles for each gene, one from each parent, and they may be the same or different. 3 things. 1. Every gene has a fixed position. Alleles are the variants found there. 2. You have 2 alleles for each gene. 3. Alleles can be dominant or recessive. Dominant alleles show up even with 1 copy, recessive only when you have 2 of them. A common slip. Don't confuse gene and allele. The gene is the position, the allele is the variant. There may be many alleles of a single gene across a population, but any individual carries only 2. Quick example, the eye color gene has alleles for brown, capital B dominant, and blue, lowercase b recessive. Possible genotypes, capital B, capital B, capital B, small b, small b, small b. Each person carries one of these 3 combinations for the eye color gene. 2 alleles total, 1 from mum, 1 from dad. Hello and welcome to pH Biology. Today's explainer is genotype, the alleles you carry for a particular gene. By the end of this video, you'll know how to write a genotype, what homozygous and heterozygous mean, and how genotype differs from phenotype. Genotype is the combination of alleles an individual has for a particular gene. 2 letters, 1 from each parent. Capital letter for the dominant allele, lowercase for the recessive. Homozygous means same alleles, like capital B or lowercase b. Heterozygous means different alleles, like capital B, lowercase b. The genotype is the genetic recipe. 3 things. 1. Genotype is written as 2 letters, 1 for each allele. 2. Homozygous means you have 2 identical copies, i.e. capital B or lowercase b. Heterozygous means you have 1 of each, i.e. capital B, lowercase b. And 3, genotype determines phenotype, but 2 different genotypes, for example, capital B and capital B, lowercase b, can give the same phenotype. Common slip. Genotype is the alleles. Phenotype is what's shown. Don't write brown eyes as a genotype. Write capital B, lowercase b, or capital B. And don't write a single letter. Genotype always involves a pair of alleles, 1 from each parent. Quick example, eye color, capital B, homozygous dominant, brown eyes. Capital B, lowercase b, heterozygous, brown eyes. The dominant capital B masks the recessive lowercase b. And lowercase b, homozygous recessive, blue eyes. So 3 possible genotypes, only 2 phenotypes. The genotype tells you more than the phenotype reveals. Hello and welcome to pH Biology. Today's explainer is phenotype, the observable characteristic that results from your genotype. By the end of this video, you'll know how genotype maps to phenotype, why dominant alleles show up so often, and where environment fits in. Phenotype is the observable characteristic an organism shows as a result of its genotype, and sometimes the environment. Dominant alleles produce their phenotype with just one copy. Recessive alleles only produce theirs when present in two copies, homozygous recessive. Different genotypes can give the same phenotype. Three things. 1. Phenotype is what you see, not what you have. 2. For a gene with one dominant and one recessive allele, there are only two possible phenotypes, but three possible genotypes. 3. For traits like height or skin color, the environment also affects the phenotype, not just the genes. Common slip. Don't write a genotype where a phenotype is asked. Brown eyes is a phenotype. Capital B, lowercase b is a genotype. And remember that environment matters for many traits. Identical twins have the same genotype, but they can have different heights if they grow up with different diets. Quick example, eye color. Genotype capital B lowercase b gives the brown phenotype because capital B is dominant. Genotype capital B also gives brown. Same phenotype, different genotype. Only genotype lowercase b gives the blue phenotype. So brown eyes covers two genotypes, but blue eyes covers only one. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download. Hello and welcome to pH Biology. Today's explainer is the dominant allele, the one that shows up in the phenotype even with just one copy. By the end of this video, you'll know exactly what dominant means and what it doesn't mean, and how to spot dominant traits. A dominant allele produces its phenotype even when only one copy is present in the genotype. Written as a capital letter, like capital B for brown, the dominant allele masks the recessive allele in a heterozygous genotype. So genotypes capital B and capital B lowercase b both give the dominant phenotype. Three things. 1. Capital letter equals dominant. 2. A single copy is enough to show the trait. 3. Dominant doesn't mean common, strong, or better. It just means it masks recessive alleles. A dominant allele can still be very rare in a population. Common slip. Dominant doesn't mean most common in the population. It just means that one copy is enough to show the trait. The dominant allele for polydactyly, i.e. extra fingers and toes, is actually rare. Most people don't carry it. Quick example, polydactyly is dominant. A single capital P allele is enough. So genotypes capital P and capital P lowercase p both have polydactyly. Only lowercase b has the normal number of digits. So capital P lowercase b parent crossed with lowercase p parent gives a 50-50 chance of each child having polydactyly. Hello and welcome to pH Biology. Today's explainer is the recessive allele, the one that only shows up when you have two copies. By the end of this video, you'll know what carrier means, why recessive diseases skip generations, and how to hand a Punnett square. A recessive allele only produces its phenotype when two copies are present, when the genotype is homozygous recessive. Written as a lowercase letter, like lowercase b for blue, if a dominant allele is also present, it masks the recessive allele. So only the genotype lowercase b gives the recessive phenotype. 3 things. 1. Lowercase letter equals recessive. 2. You need 2 copies for the trait to show. 3. Heterozygous individuals, i.e. capital B, lowercase b, are carriers. They have 1 recessive allele, but don't show the recessive phenotype. They can pass the recessive allele to their children. Common slip. Recessive doesn't mean rare or weak. It just means the allele is masked by a dominant one. Cystic fibrosis is recessive. A single recessive allele doesn't cause it. You need to inherit the recessive allele from both parents to have the condition. Quick example. Cystic fibrosis is autosomal recessive. 2 carrier parents, capital F, lowercase f, crossed with capital F, lowercase f. A Punnett square gives 1 capital F, 2 capital F, lowercase f, 1 lowercase f. The lowercase f individual, i.e. 1 in 4, has cystic fibrosis. The 2 capital F lowercase f individuals are healthy carriers. They don't have the disease, but can pass it on. Hello, welcome to pH Biology. Today's explainer is mutation, a change in the DNA sequence. By the end of this video, you'll know what causes mutations, why most have no effect, and how a rare beneficial mutation can drive evolution. A mutation is a change in the sequence of bases in DNA. Mutations happen all the time, sometimes by random copying errors, sometimes by exposure to mutagens like UV radiation or chemicals. Most mutations have no effect. Some are harmful. A rare few are beneficial, and these drive evolution. 3 things. 1. Mutations happen at random. 2. Most occur in non-coding DNA or are silent, having no effect on the phenotype. 3. Beneficial mutations give individuals a survival advantage. They reproduce more, pass on the mutation, and over many generations the population evolves. Natural selection in action. Common slip. Mutations aren't directed towards what an organism needs. They're random. The environment then selects which random mutations are beneficial, and mutations aren't always bad. Without them, no genetic variation, no evolution. Quick example. A random mutation in a bacterium gives it resistance to an antibiotic. When that antibiotic is then used widely, non-resistant bacteria die, and the resistant one survives, reproduces rapidly by binary fission, and within hours the population is dominated by resistant bacteria. Natural selection happens in real time. Right. If any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download. Hello and welcome to pH Biology. Today's explainer is homeostasis, keeping the internal conditions stable in the face of constant change outside. By the end of this video, you'll know what's regulated, why it matters, and how negative feedback keeps everything in range. Homeostasis is the regulation of internal conditions of the body to maintain a stable internal environment in response to external and internal changes. Body temperature, blood glucose, water balance, blood pH, all kept within tight ranges. Homeostasis is essential because cellular processes, especially those involving enzymes, only work in a narrow range. 3 things. 1. Multiple variables are regulated. Temperature, about 37 degrees centigrade. Blood glucose, about 90 milligrams per deciliter. pH, about 7.4. Water and iron balance. 2. Control involves receptors, a coordination center, and effectors. 3. The mechanism is negative feedback. Change is detected and the body responds to reverse it. Conditions return to the set point. Common slip. Homeostasis isn't about staying constant. Conditions fluctuate within a tight range, and it's not one system. It involves the nervous system, the hormonal system, and many organs working together: the brain, the pancreas, the kidneys, the skin, the liver. Quick example. Your body holds core temperature within a degree of 37. Walk outside in freezing weather, vasoconstriction, hair erection, shivering kick in. Walk into a hot room, vasodilation, sweating. Both responses are negative feedback, reversing the deviation from 37 degrees. Hello and welcome to pH Biology. Today's explainer is negative feedback, the control mechanism behind every homeostatic system in your body. By the end of this video, you'll know exactly how it works, the 3 components every feedback loop needs, and where you see it day to day. Negative feedback is a mechanism where a deviation from a set point triggers a response that returns the system back to the set point. 3 components. A receptor detects the change, a coordination center processes the information, and an effector carries out the response. Once normal is restored, the response stops. 3 things. 1. 3 components: receptor, coordination center, effector. 2. The response reverses the change. 3. When conditions return to normal, the response shuts off. Without that off switch, the system would overcorrect in the opposite direction. Common slip. Negative feedback reverses deviations. Positive feedback would amplify them, which is why positive feedback is rarer and usually dangerous in living systems. Negative feedback keeps things stable. Positive would spiral out of control. Quick example: blood glucose rises after a meal. Beta cells in the pancreas, the receptor, detect the rise. Insulin, the response, is released. Liver and muscle cells take up glucose. Blood glucose falls back to normal. Insulin secretion stops. Beautifully balanced. Same logic for temperature, water balance, and blood pH. Hello and welcome to pH Biology. Today's explainer is the Reflex Arc, the pathway of a reflex action, bypassing the brain for speed. By the end of this video, you'll know the sequence, why it's faster than conscious thought, and a real-life example you can feel. A reflex arc is the pathway of a reflex action: stimulus, receptor, sensory neuron, relay neuron in the spinal cord, motor neuron, effector, response. It doesn't involve conscious thought. The signal only goes as far as the spinal cord before triggering the response, much faster than going up to the brain and back. 3 things. 1. The pathway is fixed. Stimulus to receptor, to sensory neuron, to relay neuron in the spinal cord, to motor neuron, to effector. 2. The brain isn't involved in the rapid response, though it's notified afterwards. 3. Synapses connect each pair of neurons, with neurotransmitters crossing each gap. Common slip. Reflex arcs do involve the spinal cord, just not the brain. The brain registers what happened after the response. That's why you feel pain a moment after pulling your hand away from a hot pan. The response came first, the awareness second. Quick example: hand on hot stove. The temperature receptors in the skin fire. Sensory neuron sends impulse to spinal cord. Relay neuron passes it to motor neuron. Motor neuron triggers the arm muscle to contract. Hand pulled away. Total time, about 1/10 of a second. The brain registers the pain a moment later. Hello and welcome to PH Biology. Today's explainer is the synapse, the gap between two neurons crossed by chemical messengers. By the end of this video, you'll know why electrical signals become chemical, why synapses are slower than the rest of the nerve, and how neurotransmitters work. A synapse is the junction between two neurons. When an electrical impulse reaches the end of one neuron, it triggers the release of a chemical neurotransmitter into the synaptic gap. The neurotransmitter diffuses across the gap and binds to receptors on the next neuron. If enough binds, a new electrical impulse starts in the second neuron. Three things. 1. Transmission across the synapse is chemical, not electrical. 2. Synapses are slower than transmission along a neuron because diffusion takes time. 3. Synapses allow signals to be regulated. They can be amplified, dampened, or modulated by drugs and medications. Common slip. Don't say the signal jumps across the synapse. It's carried by neurotransmitter molecules diffusing across, and synapses are slower than along a neuron, which is why reaction times have a measurable minimum, usually around 1/5 of a second. Quick example, the ruler drop test for reaction time. Catch a ruler with your fingers. Average time around 0.2 seconds. Most of that is synaptic delay. Multiple synapses between the sensory neurons, the eye, the brain, and the motor neurons, the fingers. The electrical conduction along the nerves is much faster. Hello and welcome to PH Biology. Today's explainer is the hormone, a chemical messenger that travels in the blood from an endocrine gland to its target. By the end of this video, you'll know what makes a hormone different from a neurotransmitter, the main endocrine glands, and where hormones win out. A hormone is a chemical messenger released into the blood from an endocrine gland. It travels in the bloodstream to its target organ and produces a response. Slower than nerves, but the effects last longer and reach wider areas of the body. Each hormone has specific target cells with matching receptors. Three things. 1. Hormones travel in blood, not along nerves. 2. They're slower than neural signals, but the effects last longer. 3. Only cells with the right receptor respond. Insulin only acts on cells with insulin receptors, liver, muscle, fat cells. Common slip. Hormones and neurotransmitters are different. Both are chemical messengers, but hormones travel in blood. Neurotransmitters cross synaptic gaps. And the main endocrine glands you must know, the pancreas, the thyroid, the adrenal, pituitary, ovaries, and testes. Quick example, eat a meal. Blood glucose rises. The pancreas releases insulin into the blood. The insulin reaches the liver and muscle cells with insulin receptors and triggers glucose uptake. Slower than a reflex. Takes minutes, not milliseconds, but the effect is sustained and reaches every cell that needs it. Hello and welcome to PH Biology. Today's explainer is insulin, the hormone that lowers blood glucose. By the end of this video, you'll know where it's made, what it does, and why type 1 diabetics need to inject it. Insulin is a hormone produced by the beta cells in the pancreas. It's released when blood glucose is high, usually after a meal. Insulin causes liver and muscle cells to take up glucose from the blood. It also stimulates the liver to convert excess glucose into glycogen for storage. And the result, blood glucose falls back to normal. Three things. 1. Insulin lowers blood glucose. Glucagon raises it. Don't confuse them. 2. Insulin works on liver and muscle cells primarily. 3. Insulin is a protein hormone, destroyed by stomach acid if swallowed, which is why type 1 diabetics inject it rather than take a pill. Common slip. Insulin lowers blood glucose. Type 1 diabetics inject insulin before meals to handle the glucose surge from food. Type 2 diabetics still produce some insulin, but their cells respond poorly to it, insulin resistance, managed by diet, exercise, and sometimes medication. Quick example. Eat a big plate of pasta. Blood glucose rises within minutes. Beta cells in the pancreas detect the rise and release insulin. Liver and muscle cells take up glucose. Liver converts the excess to glycogen. Within an hour, blood glucose has returned to normal. Insulin secretion stops. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download, plus the master. Hello and welcome to PH Biology. Today's explainer is glucagon, the hormone that raises blood glucose. By the end of this video, you'll know how it differs from insulin, where it's made, and what happens during a long workout or between meals. Glucagon is a hormone produced by the alpha cells in the pancreas. It's released when blood glucose is low. Glucagon causes the liver to break down stored glycogen back into glucose, which is then released into the blood. The result, blood glucose rises back to normal. 3 things. 1. Glucagon raises blood glucose, insulin lowers it. They're opposite hormones, both made by the pancreas by different cell types. 2. Glucagon works on the liver, mobilizing stored glycogen. And 3. Without glucagon, your brain would have no way to keep glucose available between meals. Common slip. Glucagon and glycogen are easily confused but different. Glucagon is a hormone. Glycogen is a storage molecule, a long chain of glucose units. Glucagon causes glycogen to break down back into glucose. Quick example, long distance run. 2 hours in, your blood glucose is dropping fast. Alpha cells in the pancreas detect the fall and release of glucagon. Glucagon tells your liver to break glycogen back to glucose. Glucose pours into your blood, fueling your muscles. Glucagon secretion stops when the blood glucose returns to normal. Hello and welcome to PH Biology. Today's explainer is vasodilation, the widening of blood vessels near the skin surface to help the body lose heat. By the end of this video, you'll know exactly when and why it happens and what it pairs with to cool you down. Vasodilation is the widening of blood vessels, particularly those supplying the skin, to allow more heat to be lost from the body. Triggered when the body is too hot. More blood flows close to the skin surface and more heat radiates away to the cooler air around you. Pairs with sweating to bring the temperature down. 3 things. 1. Vasodilation opens up vessels. Vasoconstriction is the opposite. 2. It happens when the body is too hot. 3. Sweating works alongside vasodilation. Sweat evaporates from the skin, taking heat away through evaporation. Both responses are triggered by the hypothalamus. Common slip. Vasodilation doesn't add blood to your skin. It just lets more of your existing blood flow through wider vessels. Cardiac output is the same. It's just the distribution changes. And don't say blood vessels dilate. Say vasodilation occurs or blood vessels widen. Quick example, hot summer day. Skin looks flushed. That's vasodilation at work. Sweat dripping down your face, evaporation cooling you. You feel hot, but your core temperature stays near 37. Without vasodilation, heat would build up in your core and core enzymes would denature. Hello and welcome to PH Biology. Today's explainer is the ecosystem, the interaction of a community of living organisms with their non-living environment, By the end of this video, you'll know the boundary between biotic and abiotic and how interdependence holds everything together. An ecosystem is the interaction of a community of living organisms, biotic factors, with the non-living parts of their environment, the abiotic factors. Ecosystems range from tiny, a pond, a faunan log, to enormous, a rainforest, the entire ocean. All ecosystems share the same logic. Living things depend on each other and on their environment. Three things. 1. Biotic is equivalent of living. Abiotic is the equivalent of non-living, i.e. light, temperature, water, soil, pH. 2. Interdependence. Every organism depends on others and on abiotic conditions. 3. Disturb any one factor and the rest feel it. Lose the bees, lose the flowers. Acidify a pond, lose the fish. Common slip. An ecosystem isn't just the animals or just the plants, it's the whole system, living and non-living together. And ecosystems have boundaries that ecologists choose. There's no fixed size. Quick example, a pond ecosystem. Fish, frogs, algae, water boatmen, dragonflies, decomposers in the sediment, these are all biotic. Plus, water, dissolved oxygen, light, temperature, pH, soil at the bottom, abiotic. Every component depends on the others. Heat a pond up by 5 degrees, oxygen levels drop, fish struggle, algae bloom, ecosystem changes. Hello and welcome to pH Biology. Today's explainer is the food chain, a sequence showing how energy passes from one organism to the next through eating. By the end of this video, you'll know the trophic levels, why arrows point a specific way, and why food chains are rarely longer than 4 or 5 steps. A food chain shows the sequence of who eats whom in an ecosystem, with arrows showing the direction of energy flow. Each level is a trophic level. Producers make their own food via photosynthesis. Primary consumers eat producers. Secondary consumers eat primary. Tertiary consumers eat secondary. 3 things. 1. About 10% of energy is transferred between trophic levels. The rest is lost as heat in waste or as parts not eaten. 2. That's why food chains are rarely longer than 4 or 5 levels. 3. Arrows show the energy flow into the next organism, not what eats what. Common slip. Arrows go into the consumer, showing the energy flowing into them. Don't draw them the other way. And remember, producers are almost always green plants or algae. They're the start of every food chain on Earth. Quick example, grass arrow, rabbit arrow, fox arrow, eagle. Grass is the producer, rabbit is the primary consumer, a herbivore. Fox is the secondary consumer, a carnivore. Eagle is the tertiary consumer, an apex predator. Each arrow loses about 90% of the energy as heat, waste, and uneaten material. Hello and welcome to pH Biology. Today's explainer is biodiversity, the variety of different species in an ecosystem. By the end of this video, you'll know why it matters, what threatens it, and what conservation actually means. Biodiversity is the variety of different species of living organisms in an ecosystem. High biodiversity means many different species. Low biodiversity means few. High biodiversity tends to give a more stable ecosystem. Different species fulfill different roles, so the loss of any one species has less catastrophic knock-on effects. 3 things. 1. Biodiversity is about variety, not numbers of organisms. 2. Threats include deforestation, pollution, overfishing, climate change, monoculture agriculture. 3. Conservation tools include nature reserves, breeding programs, seed banks, fishing quotas, and international agreements like cities on protecting endangered species. Common slip. Biodiversity isn't the total number of individuals, it's the variety of species. 10,000 individuals of 5 species has lower biodiversity than 1,000 individuals of 50 species. Variety matters more than count. Quick example. The Amazon rainforest has the highest biodiversity on Earth. Millions of species across plants, insects, mammals, fungi. A monoculture wheat field has very low biodiversity. Almost all one species plus a few weeds. Both are farmland or wilderness, but biologically they're worlds apart. Hello and welcome to pH Biology. Today's explainer is the producer, the organism that makes its own food using sunlight, anchoring every food chain on Earth. By the end of this video, you'll know what producers are, what they do, and why every other trophic level depends on them. A producer is an organism that makes its own food from carbon dioxide and water using light energy, i.e. photosynthesis. Almost always green plants or algae. They convert light energy into chemical energy stored in glucose. Producers are at the base of every food chain. Without them, no other trophic level could exist. 3 things. 1. Producers are usually green plants or algae. Some bacteria also count. Chemosynthesis. 2. They convert light energy into chemical energy, glucose. 3. Producers form the base of every food chain. Even apex predators like eagles depend on plants via rabbits, foxes, and smaller birds. Common slip. Producers don't eat. They synthesize their own food from carbon dioxide and water using sunlight and chlorophyll. They're autotrophs, self-feeders, as opposed to consumers, which are heterotrophs. Quick example, a leaf in sunlight is the start of the food chain. The glucose it makes ultimately feeds every consumer above it. An eagle eating a fox, eating a rabbit, eating grass. The eagle's energy ultimately came from sunlight captured by the grass leaves. Hello and welcome to pH Biology. Today's explainer is the decomposer, the organism that breaks down dead matter and recycles nutrients. By the end of this video, you'll know why decomposers are vital to every ecosystem and the role they play in the carbon cycle. A decomposer is an organism that breaks down dead organisms and waste, usually a bacterium or a fungus, releasing nutrients back into the soil. Without decomposers, dead matter would accumulate forever and nutrient cycles would break down. Plants need recycled nitrogen, phosphorus, and other minerals to grow. 3 things. 1. Decomposers are usually fungi and bacteria. 2. They release nutrients, such as carbon, nitrogen, phosphorus, back to the soil for plants to reabsorb. 3. They release carbon dioxide back to the air through their own respiration, completing the carbon cycle. Common slip. Decomposers don't eat in the usual sense. They secrete enzymes onto dead matter, breaking it down externally and absorb the broken down food. And they're a vital trophic level, not an afterthought. Quick example, a fallen log on a woodland floor. Fungi and bacteria slowly digest it over years. Cellulose, lignin, and the proteins in the wood are broken back down into carbon dioxide, water, and mineral nutrients. The carbon dioxide returns to the air for photosynthesis. The nutrients return to the soil for the next generation of plants. Hello and welcome to pH Biology. Today's explainer is the pathogen, an organism that causes disease. By the end of this video, you'll know the 4 types, how they spread, and why each type needs a different approach to treatment. A pathogen is an organism that causes disease. 4 main types: viruses, like HIV and influenza; bacteria, like tuberculosis and salmonella; fungi, like rose black spot and athlete's foot; and protists, like plasmodium, which causes malaria. Each type causes disease in different ways and is fought by the body differently. Three things. 1. 4 pathogen types: viruses, bacteria, fungi, protists. 2. Spread by direct contact, droplets, contaminated water or food, vectors like mosquitoes or shared needles. 3. Different pathogens cause different symptoms and need different treatments. Antibiotics for bacteria, antivirals for viruses. Common slip. Not all microbes are pathogens. Most bacteria are harmless or even beneficial. Gut bacteria are essential for digestion, for example, and viruses aren't really alive in the conventional sense. They need a host cell to reproduce. Quick example: flu, a virus spread by droplets in coughs and sneezes. Tuberculosis, a bacterium, also spread by droplets. Athlete's foot, a fungus spread by direct contact in damp areas. Malaria, a protist spread by mosquitoes' bites. 4 diseases, 4 pathogen types. Hello and welcome to PH Biology. Today's explainer is the antibody, a specific protein produced by white blood cells that locks onto a specific antigen on a pathogen. By the end of this video, you'll know how antibodies work, why they're specific, and how memory cells give immunity. An antibody is a protein produced by white blood cells, specifically B lymphocytes, that binds to specific antigens on pathogens. Each antibody is shaped to one particular antigen, like a lock and a key. Binding clumps pathogens together for easier removal, labels them for destruction by other immune cells, or neutralizes them directly. 3 things. 1. Antibodies are proteins, not cells. They're made by white blood cells. 2. Each antibody is specific to one antigen, different antibodies for different pathogens. 3. Once you've had a pathogen, the body retains memory cells that can quickly make antibodies again. That's immunity. Common slip: antibodies are proteins. Don't say antibody cells, and remember that antigen is the marker on the pathogen, while antibody is the protein your body makes against it. The two are easily confused and not the same. Quick example: measles infection at age 5. Your body produces antibodies and memory cells specific to measles antigens. At age 40, you're exposed to measles again. Memory cells respond fast, antibody levels surge, and the virus is destroyed before you feel ill. You're immune. Hello and welcome to PH Biology. Today's explainer is the antitoxin, a specific protein that neutralizes toxins released by bacteria. By the end of this video, you'll know how antitoxins differ from antibodies, why some bacterial diseases need antitoxins specifically, and how vaccines exploit this. An antitoxin is a protein produced by white blood cells that binds to and neutralizes toxins released by bacteria. Specific to one toxin, the same lock and key principle as antibodies. The difference: antibodies label or neutralize pathogens themselves. Antitoxins neutralize the poisons that bacteria release. 3 things. 1. Antitoxins target toxins, chemicals, not the bacteria themselves. 2. The same lock and key specificity as antibodies. And 3. Important for diseases where bacteria release damaging toxins. even if the bacteria themselves are killed by other means. Common slip. Antitoxins aren't antibodies, even though both are proteins made by white blood cells. Antibodies bind to pathogens or their antigens. Antitoxins bind to toxins, chemicals released by bacteria. Quick example, tetanus is caused by a bacterium that releases a toxin damaging nerves. Even if the bacteria are killed, the toxin can still cause paralysis. Antitoxin neutralizes the toxin. That's why a tetanus vaccine contains the inactivated toxin, training the body to produce antitoxin in advance. Hello and welcome to pH Biology. Today's explainer is phagocytosis, when a white blood cell engulfs and digests a pathogen. By the end of this video, you'll know how it works, why it's non-specific, and where pus comes from. Phagocytosis is the process by which a white blood cell, a phagocyte, engulfs and digests a pathogen. The phagocyte surrounds the pathogen, takes it inside the cell, and breaks it down with digestive enzymes. Non-specific. Phagocytes attack any foreign body, regardless of which pathogen it is. Three things. 1. Phagocytes don't need a specific antibody response to act. They're a first-line internal defense. 2. They work within minutes of infection. 3. Phagocytosis is one role of white blood cells. The other two are producing antibodies, B lymphocytes, and producing antitoxins. Common slip. Phagocytes aren't the same as B lymphocytes. Phagocytes engulf pathogens. B lymphocytes make antibodies. Two different white blood cell types, two different roles in immunity. Quick example, you cut your finger and bacteria enter. Within minutes, phagocytes arrive and engulf them. Digestive enzymes inside the phagocyte break the bacteria down. Pus at a wound site is mostly dead phagocytes plus broken down bacteria. That's your immune system at work. Hello and welcome to pH Biology. Today's explainer is vaccination, training the immune system to fight a pathogen before you've ever met it. By the end of this video, you'll know what's in a vaccine, how it produces immunity, and why some require boosters. Vaccination is the introduction of dead, weakened, or modified pathogens, or their antigens, into the body to stimulate an immune response without causing disease. The body produces antibodies and memory cells specific to that pathogen. On future exposure, memory cells respond quickly, destroying the pathogen before symptoms appear. Three things. 1. Vaccines contain dead or weakened pathogens or just their antigens. 2. They trigger antibody production and memory cell formation. 3. Memory cells stay for years, sometimes life, giving long-lasting immunity. Some vaccines need boosters to top up memory cells over time. Common slip. Vaccines don't cure disease, they prevent it. Antibiotics treat existing bacterial infections. Vaccines train the body before infection. Two different strategies, both vital. Quick example, the measles, mumps and rubella vaccine, MMR, introduces weakened forms of all three viruses. Your body produces antibodies and memory cells against each. Years later, if you encounter the real virus, memory cells respond quickly and you don't fall ill. You're immune for decades. Hello and welcome to pH Biology. Today's explainer is herd immunity, when enough of a population is vaccinated that the disease can't spread, protecting even those who haven't been vaccinated. By the end of this video, you'll know how it works and why anti-vaccine choices affect everyone. Herd immunity is the protection given to a population when a high proportion is immune to a disease. The pathogen cannot spread effectively, and even unvaccinated individuals are protected. Different diseases need different vaccination rates. Measles is highly contagious, needs around 95% coverage. Polio needs around 80%. 3 things. 1. Herd immunity protects everyone in the community, including those who can't be vaccinated for medical reasons, such as newborns or immunocompromised. 2. Different diseases have different thresholds. 3. Drop below the threshold and outbreaks return, as has happened with measles in recent years when vaccination rates dropped. Common slip. Herd immunity isn't just personal protection, it's a community effect. Choosing not to vaccinate doesn't just put yourself at risk, it puts others at risk too, particularly those who genuinely can't be vaccinated. Quick example, smallpox was eradicated globally in 1980 by a sustained vaccination campaign. Herd immunity at a planetary scale. Polio is nearly there. Where vaccination rates drop, herd immunity collapses and outbreaks return. Measles outbreaks across Europe in the 2010s followed exactly this pattern.